Living things are unique, aren't they? For instance, how are there countless generations of a certain type of species, and yet they're all different from one another?
How does a living thing's skin regenerate after being wounded? Who or what exactly are behind those damage repairs? Well, you might have guessed correctly.
The culprits who are behind these actions are body cells. Each of our body cells undergoes several consecutive processes along their lifetime, which ultimately result in allowing us to stay hopeful, go forward, and strive to keep on living.
Throughout the whole cell cycle, cells experience three main stages in which they grow and divide respectively and repetitively.
The process by which they grow larger is referred to as an interphase.
Where see the next stage is going to go through is referred to as mitosis and cytokinesis. Okay, let's first talk at the first stage of the cell cycle, interphase.
Interphase is often considered to be the preparation stage.
This referred as each cell actually gets itself ready to be divided in the following stages. It is during this stage that a cell spends most of its lifetime.
To make things more comprehensive, the very process of interphase is further divided into three separated phases.
Among them being G1, aka gap 1, S, aka synthesis, and G2, aka gap 2. During G1, a cell increases in its size due to the duplication of its internal organelles, excluding chromosomes. Most cellular activities happen amidst this phase.
On the S phase, or known as the synthesis phase, chromosomes begin replicating to form sister chromatids. The overall purpose of the particular phase itself is to synthesize or replicate the DNA. Fun fact in animal cell, the duplications of centromeres are also conducted.
Last is G2 phase. In this phase, the cellular contents of each cell is examined with the intention to make sure there won't be any needed repairs before the cell get divided. If there is in fact a detected flaw, the cell will then either be in a state of limbo until it can fix the issue, or they do apoptosis, where how do I explain this? Okay, this is a cell and this is apoptosis. Yeah, they self-destruct.
After going through interphase,
Mitosis occurs. Although it is a minor part of the cell cycle and it occurs at such a short span, mitosis actually plays a large role in our lives.
After all, the purpose of mitosis itself points towards growth, as well as damage repair. While the process itself is considered as one, most scholars divide it into four different phases.
These are prophase, metaphase, anaphase, and telophase.
Okay, let's visualize the process with this cell.
Hey look, I became a centrosome.
And I became a nuclear membrane.
Oh hey, I become a chromatin.
And these are the other organelles. Let's begin with prophase.
Oh hey, I am moving.
First, the centrosomes will be transported to the opposite ends of the nucleus.
Then, chromatin will condense into discrete chromosomes.
Lastly, the nucleus and the nuclear membranes will fade.
Did that just disappear?
Moving on with metaphase. Wow, that came out of nowhere.
The spindle fibers, which originate from microtubules, are fully formed by centrioles. After a while, they will stick into the kinetochore of the chromosomes, scattered by the pair of chromatids positioned in the middle of the spindle fiber form.
Alright, moving on with anaphase. The chromatids will be pulled by the spindle fiber to the opposite end of the poles. Last but not least, when the chromatids have reached the opposite end of the poles, it will be formed back into chromatin.
Last but not least, telophase. Once the spindle fibers have disappeared, nucleus will be formed back and cytokinesis will occur in this phase. This process is known as the division of cytoplasm, where the cell formation will be halved and divided into two, equal size and shape, into two daughter cells. A good example of the process of mitosis can be seen or observed when our skin gets wounded. After a short few while, slowly but surely, our skin cells will be repaired. The process by which our skin cells get repaired is called mitosis.
Meiosis plays a significant role in the cycle of cell division, since it's a process that contributes to genetic variety.
Unlike mitosis, it will undergo the process in two different stages, characterized by process 1 and 2.
Stage 1 has prophase 1, metaphase 1, anaphase 1, telophase 1, while stage 2 has prophase 2, metaphase 2, anaphase 2, and telophase 2.
Alright, let's imagine a cell again. Hey look, now I became a centrosome.
And now, I have become a nuclear membrane.
Why am I still a chromatin?
Now, let's see prophase 1. Unlike the prophase process performed in mitosis, meiosis has 5 stages of prophase,
which are leptotin, zygotin, pachytin, diplotin, and diakinesis. Now, let's start with leptotin. Chromatin is converted into chromosomes in the leptotin stage.
In zygotin stage, tetrad and bivalent is formed. This is where two chromosomes stick together by the help of synaptonymal complex.
Next is the pachytin stage. This is where the chromosomes perform an action called crossing over. When the chromosomes cross over, chiasma is made.
Last is diakinesis. In this stage, the nuclear membrane will disappear.
Now we're gonna start with metaphase 1. Like mitosis, there will be spindle fibers. The only difference is that the bivalent will be sorted in the middle now.
Now come to anaphase 1. Like mitosis, the spindle fiber will pull the centrosomes.
Different from mitosis, the whole chromosomes will be pulled by the spindle fibers to the opposite end of the poles. Last but not least, telophase 1. Through this phase, nuclear membrane will appear again, followed by the emergence of a contractile ring. Cytokinesis will occur, and the cell will then split into two equal-shaped cells, each containing half the amount of chromosomes in comparison to its original.
Different from mitosis, the total chromosomes in stage 1 of meiosis will be half the total of the chromosomes in mitosis, which is why meiosis is important in genetic variety.
Prophase 2. The nuclear membrane also disappear, yet this time the chromosomes are already present. Thus, there is no need for condensation anymore.
During metaphase 2, spindle fibers will be created from the centrosomes and the chromosomes will be organized as a line in the middle.
In anaphase 2, the spindle fibers will carry the chromatids.
In telophase 2, nuclear membrane will appear again, and with the help of contractile ring, cytokinesis will occur, but this time the chromatid don't change into chromatin.
Eventually, the implication of meiosis is the creation of 4 daughter cells. An example of meiosis can be observed when the sperm from a male reproductive system meets with an egg cell from a female reproductive system.
Mitosis and meiosis are both classified as cell division. However, there are several differences in the processes.
Mitosis will produce 2 identical daughter cells with equal amounts of chromosomes, or it is known as diploid. Meanwhile, meiosis will produce 4 daughter cells with half the amount of chromosomes, or known as haploid. This is because while mitosis undergoes one phase, meiosis undergoes 2 phases of the cell division.
There also exist several differences in the prophase process. While in mitosis, the prophase process took shorter to occur, prophase 1 has already 5 stages to begin with in meiosis. In addition with prophase 2, which is basically the same as the prophase process belonging to mitosis.
While crossing over occurs in prophase 1 of meiosis, no such thing exists in the prophase process of mitosis. This is due to the very purpose of mitosis, cell division, which is generally aimed for growth and cell repairs. This is different from meiosis, however, in which its main purpose is actually the production of genetic diversity of descendants. Such, of course, can be achieved through sexual reproduction, and yet, meiosis plays its part by allowing the production of gametes to be established.
To conclude, all living things wouldn't have been able to live today in the absence of interphase, mitosis, as well as meiosis. We believe that the comprehension of those three processes can help us understand more health-related aspects in our daily lives.
This is especially true for those of us who are interested in learning the secrets of our body's wound coverage.
That being said, scientists are still discovering better ways to empower our body functions, raise its regeneration efficiency, as well as to increase its gamete establishment rate. As of now, we can only wait to witness what the future holds.